Good morning, Zhao Shang Hao. Today, I'll be talking to you about our recent work on taurine as a driver of aging. It is my pleasure to be presenting to you our work that lasted more than a decade. So, human society is aging. We have been told since the day we are born that we are going to age one day and die. Hopefully, during the course of my talk, I can convince you that this process can be modified, and today we can manipulate it through variety of means to extend a healthy lifespan. So the aging predisposes us to variety of chronic diseases such as osteoporosis, cancer, cardiac diseases, cognitive decline, so on and so forth. And aging remains one of the greatest predisposing factor for these diseases. Therefore, if we can target the basic biology of aging, we have a means to delay the aging population. And in other words, we can increase the health of the population, suppress these morbid diseases that happen at the end of our lives. My lab investigates how molecular composition of organisms changes with age to identify novel interventions to target age-related diseases. An organism is made up of different molecules. They can be derived from nutrients, they can be produced within our body, and then we classify them as semi-essential, or when they are obtained only from the diet, they become essential. Now, the importance of these molecules is very well emphasized in the literature in the human society itself. If we look at now just 100 years, last 100 years, one can notice that many of the molecules were missing in human diet, and they were later on classified from 1920s onwards as vitamins, starting from vitamin B1, A, C, and so on and so forth. Now, today I'll talk to you about one such molecule, a semi-essential molecule that is derived from the diet. Now, in our quest to identify the molecules that are regulated during the aging process and that have a potential to affect the aging process, we performed a metabolomic screen in aged humans way back in 2012. This screen showed that one of the most dramatically down-regulated molecule in this screen was taurine. So let us find out. Now, taurine is a atypical amino acid. It contains a sulfonyl group in place of a carboxyl group present in typically in other amino acids. And it has an amine group. That is why it is known as an amino acid. It was identified way back in 1827 by two German scientists, Tiedemann and Gmelin. For almost 140 years, taurine remained in hibernation because molecular biology and biochemistry was just evolving. Due to the increase in the affluence of society in post-World War II, there was increased demand for pet foods in the society. Because people had now 9 to 5 jobs, they had to provide some kind of a food to their pets when they went for a job in the morning. And if they had those pellets, when they came back home, these pets were happy. One of the most striking aspect of this introduction of pet foods in the society was development of variety of diseases in these pets, such as growth retardation, cardiovascular abnormalities, obesity, and one important event that was noted by the society during this introduction of pet foods was that cats started going blind. They were visibly growth retarded and blind. This led to increased investment in science and technology to identify the reason for the blindness in cats. The investigation in this process, which lasted around 15 to 20 years, led to the discovery in 1975 that retinal degeneration or blindness in the cats is caused by a single amino acid deficiency in their diet, and that amino acid was taurine. Since then, cat foods have been supplemented with taurine, and blindness and growth retardation have overall gone away. Now, when we were starting, it was already known that taurine levels in the blood are associated with variety of health parameters. However, it was not known whether these taurine levels affect the aging process. And that is what we set out to do. Now, to begin with, we measure taurine levels in the blood of mice during the aging. And as you can see, that taurine levels decline with the age in the mouse. When we looked at in monkeys, taurine levels declined in aged monkeys as well. In humans also, same trend followed, and taurine levels were abundance was lower in aged humans. In other words, taurine abundance declines with age in variety of species. To find out whether this decline in taurine abundance that happens with age is a driver of aging and actively participates in the aging process, or is it a passenger of aging and is just a collateral damage that happens during the aging process. To do so, we then next fed middle-aged mice around 14 months of age, which are equivalent to around 45 years old humans, with once daily taurine supplementation for life. This taurine supplementation led to increase in the median lifespan by around 12% in females. And in males, the increase was about 10%. So these results show that a reversal of taurine deficiency that declines with age in mice leads to increase in the lifespan of the animal. One of the most important aspects of any anti-aging intervention is to see whether the animals that are now living longer, are they living healthier? Because you do not want to be living longer and full of morbid diseases. When we looked at a variety of organ system functions after 1 year of once daily taurine supplementation in these aged animals, we saw that increase in muscle strength and neuromuscular coordination, increase in the bone density, and these bones were less fragile. They had a better glucose and insulin tolerance. Their GI motility was better. In the brain, they had lower anxiety, and these animals had more memory. Immune system function was now enhanced in such a manner that it actually appeared similar to younger immune system. They also had a dramatic reduction in their adiposity. And this reduction in the fat amount in their body was due to a major increase in the energy expenditure of these animals because their food intake was relatively normal. Now, these results tell you that a reversal of taurine deficiency during the aging process leads to increase in the lifespan and health span of mice. So how does taurine enter the cell and affect different organ functions? The taurine, one of the predominant transporter that is required for taurine to enter the cell is SLC6A6. Once taurine enters the cells, it performs a variety of function. Now, in order for us to establish that taurine deficiency is a driver of aging, we then next knocked out this transporter in mice. And this reduction of the taurine transporter on the cells led to dramatic reduction in the intracellular levels of taurine in a variety of tissues. This reduction of taurine or induced taurine deficiency led to increase in the variety of diseases in these animals, which were just 4 months old. And this increase in morbidity curtailed their lifespan by around 35 to 40%. So the normal median lifespan of animals was around 28 to 30 months of age. And if you look at now induced taurine deficient animals, they were living around 16, 17 months only. So taurine deficiency curtails healthy lifespan. Now, how does taurine suppress all these aging phenotypes that we were observing during the aging process? What we identified using variety of different species is that taurine, once enters the cells, it variety of downstream processes as stem cell renewal in different tissues. It increases autophagy. It maintains the proteostasis in the cells. It suppresses DNA damage. It also promotes epigenetic changes on histone and DNA. And it was suppressing senescence dramatically. One event that we noted is that taurine was actively transported into the mitochondria, where it modified a mitochondria specific to RNA, tRNA leucine, increasing the ND6 translation and promoting complex 1 activity and thereby suppressing the ROS activity in the cells. Now, together, these studies identified that in the cells, taurine affect variety of different aging hallmarks that are enriched in cells with age. And that is how it was increasing the cellular and, as a consequence, organismal functions. Now, to get closer to humans, we then looked at in 12,000 humans, for which we had a longitudinal health data available. We measured taurine, hypertaurine, a precursor of taurine, and N-acetyl-taurine. These analysis of this correlation between the health parameters and taurine and metabolite abundance showed that if humans had a higher, these are 60-year-old humans, if humans had higher taurine levels in age, they had a lower obesity, lower BMI, lower waist to hip ratio, less type 2 diabetes incidences, they had a less liver damage, their inflammatory marker CRP was lower in them, and they had lower hypertension, and so on and so forth. In other words, taurine deficiency in aged humans associates with poor health as well. Now, we next looked at, okay, what happens to taurine and its metabolite levels in a healthy state of a human being. To do so, we then put humans through a bout of exercise, sedentary subjects, natural bodybuilders, endurance athletes, or sprinters. What we identified is that the abundance of taurine and its metabolites increase in the blood of these exercising humans. So a healthy state of a human increases taurine and its metabolite levels, suggesting that perhaps some of the health benefits of exercise are mediated by increasing the taurine and metabolite levels in the bloodstream. To then demonstrate the biomedical relevance of these findings, we then performed a monkey intervention study, for which we treated aged monkeys, which are closer to 45, 50-year-old humans, with taurine once daily for 6 months and measured their health parameters non-invisibly. Taurine supplemented monkeys had lower body weight, they had lower percent fat gain, they had more bone density, bone mineral content, they had a better glycemic index upon taurine supplementation. They had a lower markers of liver damage in their bloodstream. And their immune system was functioning similar to a younger looking immune system. And their oxidative stress markers for DNA, lipid, or protein were dramatically suppressed in a variety of different assays. Now, these results show us that taurine intervention increases monkey health as well, a species that is closest to the human being. Now, to summarize, what I have shown you is that taurine abundance declines with age in several species, and once daily taurine supplementation from midlife increases the lifespan of mice. These mice that were living longer, they were also living healthier by through which we showed by measuring their health span. And these different health span measurements were as a consequence of taurine suppressing or ameliorating variety of aging hallmarks that get enriched with age in the cells. When we looked at different other species, such as lower invertebrates in C. elegans model, these also had a dose-dependent increase in the lifespan of worms, but not in the yeast. In humans, we showed that lower taurine is associated with the poor health, such as more incidence of diabetes, obesity, or inflammation. And a monkey intervention study showed us that monkeys that were supplemented with taurine had a better health parameters. So in other words, taurine deficiency is a driver of aging. Now, taurine has been shown to treat diseases. It has been shown in various clinical trials to affect liver damage, to improve blood pressure, to make humans with their less type 2 diabetes incidences. Also, it has been shown to suppress the postmenopausal symptoms. Now, this is where we are heading. We are heading towards identifying, can taurine supplementation in humans delay the pace of biological aging? Because that has not been tested till date. And the approach we are using is a randomized clinical trial and measuring variety of health span and molecular markers in humans, for which we are currently establishing the consortium of basic biologists and clinicians. Now, together, I have shown you that aging is not an inevitable process. Genetically, in different model organisms, it can be modified today. And with the lifestyle and drug interventions such as rapamycin, metformin, acarbose, senolytics, and now taurine, we have a means to now delay the process of aging or make aging a healthy venture. These results have been obtained through an awesome collaborative interdisciplinary international team. The labs have been listed on the left, and by the students and postdocs who performed the work have been listed in yellow. These results have been supported by NIH grants to myself. Thank you very much for your attention, and I will be happy to answer any questions you have.
Hi, great talk. So I have two questions. One is that you mentioned that the taurine is declining with aging, right? So if this is right, so this is due to the food. I believe that the people eating the similar food from get older, right? So then it's do you see the declining of transporter SLC6A6? It's declining also with aging. This is can explain why the taurine is reduced with aging. So this is the first question. The second one is that do you measure the taurine concentration in different organs? So is it possible that different organs have different a need for the taurine? So thank you.
Very important questions. The first one is that why does taurine abundance decline in animals? We don't know the answer for it precisely. We have the data now that shows that not only taurine synthesis within the body is perturbed with age, there is a level of taurine resistance in different tissues. And also, taurine absorption from the GI tract from the diet also is decreasing. So the decline in taurine abundance with age occurs due to a variety of factors such as synthesis, metabolism, and its action also is compromised in tissues. Now, coming to the second question, that is, are different organs differentially sensitive to taurine? You are absolutely right. Some organs can produce taurine to an extent, such as liver can produce to the most part, pancreas to some extent, and brain to some extent. But some organs don't produce taurine at all, such as muscle. So the first indication of taurine deficiency is seen in muscle wasting or muscle coordination defect or brain health defects. So this also raises that, yes, different organs are not equally sensitive to taurine deficiency because they have some ability to a limited extent producing taurine, and organs that do not produce taurine are more susceptible to taurine deficiency.
Yeah, it's a really awesome collaboration, and it's really, really wonderful finding. Only a single compound can make the animals live much longer. It's really amazing. Of course, you mainly from age depend decrease. Then you think give a supplementation, it will help. But the question like this, not only taurine, we know so many things decline with aging, like melatonin, could kill like vitamins or something. So how can only one supplementation can do all of things? This is my question. Why taurine can help others?
It is an important question. And I think the answer to that is that we have noted that taurine can promote health of cells by a variety of downstream perturbations, such as, for example, one of the most remarkable results we obtained in zebrafish, where now zebrafish that were telomerase deficient, they die with 40% of them die after birth within 10 days. Now, these telomerase deficient fishes, if you provide them taurine, they were now completely rescued with a 300 micromolar, 10 millimolar dose. What it is telling us is that taurine is able to suppress downstream consequences of various deficiencies as well. So, in other words, we have a molecule that is only one of the pieces of puzzle of aging. It is not the only one, but it can promote health of organism. I'm sure that there are many other molecules that will be discovered in going forward, which will now have an additive effect with taurine or additive effect with other interventions. And these studies, I think, will highlight the importance of these nutrients, which we have very limited understanding. Vitamins were just discovered in 1928. We knew scurvy disease, we knew pellagra, we knew beriberi, but all single nutrient deficiency was causing all these different multitude of health problems. And this also emphasizes that perhaps there is a need for us to look at how evolutionarily human diet and genetics has shaped up human health. And perhaps it is a time for us to look at, emphasize a proper nutrition, a quality nutrition, not just a nutrition and looking at calories, which are present on the variety of labels in the foods that we eat today.
Oh, this is a wonderful talk, and I have one question. It's more like a follow-up question. So we sort of see so many reports in the mice that taurine can extend life, and metformin extend life, and many other things. So is anyone in this field or planning, you put them all together? I mean, do they have any add up effect? And this one live 2 months, that one live 5 months. If they're putting 10 that are all effective, when putting 10 will be like extend to 1 year, or they are all about, once you put a 10 together, they probably just do exactly just like one, because they could be all through other mechanisms. Maybe, for example, even you said they eat normally, but they could be that regulate the absorption or something that, or through the calorie restriction effect or some common effect, that therefore they don't have cooperative or even additive effect. What's your comments?
Absolutely. I think it's a very important question. And people are now looking at combinatorial therapies. However, I must note here that combinatorial therapies are very difficult to interpret. We have done with, for example, with metformin, combination with taurine, or rapamycin with taurine. And we do see that if the downstream mechanisms are different, for example, rapamycin, which affects the mTOR pathway, mTOR complex, taurine does not affect mTOR directly. It only affects downstream processes. If we combine rapamycin with metformin, with the taurine, we are able to have a additive effect on health. Currently, these cohorts of animals are being put through the aging trials, and we would know perhaps in next couple of years whether the lifespan extension that we observe with rapamycin and taurine, is there an additive effect of these two molecules going forward. But I can tell you that rapamycin and taurine, these two molecules, we have noted that at least for few functions, they are having an additive, and in one organ function, it was synergistic effect, where it was exponential increase, not just addition of the two molecules together.
So Yadav, Steve Ostead here, very nice talk. Good to see you. I've let slightly different interpretation that one could have, which is that the diets that our mice and our monkeys are deficient in taurine, and that what we're doing is basically giving them a better diet. That seemed consistent, what you found?
You are absolutely right. I think I would not say that the diet that we feed to mice are the ideal diet. These are the animals which we bred and we modified them through genetic crosses for the last century or so. Not only animals, even for humans for that matter, we do not know what is the ideal diet for a human being. Maybe we are deficient in some molecules that we are now, not only now, I would not say now, only in the last century or so, we have been able to now bring up these molecules like vitamins, for example, B1, B3, B6. They were causing diseases in humans. We did not know about them. humans are we are ancestrally we are hunters, right? We were living in the wild in forests, and we were dependent on the diets that were more raw or natural. Now, when the domestication of the species has led to the precipitation of diseases, not only in wild animals like cats, dogs, mice, but in humans as well, because we have self-domesticated ourselves. We are not used to living in a structure like this, where these nutrients which are essential for us, we do not yet have a precise understanding of the nutrient composition that is required for ideal composition required for a perfect life. And that, I think, will be an important question for us to understand, that how can we modify the quality of diet, not only looking at these calories which we eat on the packaged diets. And I think we'll have a better understanding of the nutrient genetic interactions with the different ethnic groups or populations, which will highlight how can we now modify what nutrients are essential or important for us for a best health going in the aging process.
Thank you. Thank you, Professor Vijay, for sharing with us today. We'll be continuing with our agenda now, and we hope to hear from you soon. So goodbye for now. Thank you very much.
Thank you very much. Thank you.